The structures that are considered to be organelles, they have membranes. They are going to have their own lipid bilayer membrane. So that means this cell is not only working to create a membrane around the cell itself, it's working to create a membrane around these organelles. The nucleus is one that has the double membrane layer. So our nucleus, for our purposes, usually shown as being somewhere in the middle of the cell, it has a phospholipid bilayer. It is a double layer structure in the cell. We're going to find to the inner portion is our nucleolus. The fluid existing around it is the nucleoplasm, and they refer to this double layer membrane as our nuclear envelope. This is the area that we find our chromosomes, our DNA. DNA never leaves the nucleus of the cell. DNA, we know as our chromosomes. In an animal cell, we have 46 chromosomes. One chromosome is a DNA molecule. So that means in the nucleus of our cells, we have got 46 DNA molecules. On those molecules, they pair up and give us 23 pairs. 1 through 22 are our autosomes. Pair 23 are the sex chromosomes, XX or XY. Each strand, each molecule of DNA contains genes. Some of them tens of thousands of genes. Does that make sense? It can exist as chromatin, looking like spaghetti inside of the nucleus. When the cell needs to duplicate, and there are only two cells of the body that don't: nerve cells, muscle cells. All other cells of the body duplicate, reproduce. Before they do that, this is where the 46 pairs have to condense, and they form what we begin to look like that. That would be a pair, and they're held together by that centromere, and then they get to split, where one, so that we end up with two new cells. So everybody remember that. Okay? So they can exist as the chromatin. They can exist as the chromatids. All right? And then we usually refer to them when they pair up, 1 through 22, as our chromosomes, with pair 23 being the sex chromosomes. They are carrying our genetic material to code for everything. ribosomes inside the cell. We can have ribosomes that exist inside the cytoplasm, and they can do their job, which is protein synthesis. What is that?
Making protein.
That is making the protein. So, if I have a chromosome, okay, and if this is my chromosome made up of all those bases, A's, T's, G's, C's, okay, along this chromosome strand are areas of genes. The bases that are found, A's, T's, G's, C's, every 3 code for an amino acid. So I have an amino acid, connect to an amino acid, connect to an amino acid for however many bases are in this gene. These amino acids code for protein. The process of making this protein is protein synthesis. Synthesis, synthesize, make, okay? So ribosomes in the cytoplasm, they start out making the proteins. They can do this completely in the cytoplasm, or they can attach to the endoplasmic reticulum. Here's the difference. The free ribosomes, where the protein gets produced in the cytoplasm, that protein stays in the cell. For example, it might go to the plasma membrane. If the ribosome moves to the endoplasmic reticulum, this is making a product to go out of the cell. Free ribosomes make a protein that stays in the cell. Attached makes a product that goes out the cell. This is giving us an idea of those ribosomes. This is DNA within our nucleus. Messenger RNA reads the gene, comes out of the nucleus, attaches to ribosomes. Ribosomes either make that protein and stay in the cell, or they make a product that leaves and goes out of the cell. Ribosomes contain a small, which I tried to represent here, a small unit and a large unit. And their goal, protein synthesis. The free ribosomes make something that stays inside the cell. If the ribosome attaches to my endoplasmic reticulum, okay, it makes a product that goes out. Our endoplasmic reticulum, therefore, if ribosomes attach to it, it's called rough. But we also have a portion of endoplasmic reticulum that's considered smooth. The smooth doesn't have ribosomes attached to it. It makes lipids for the cell to use. And in some areas of the body, helps detoxify any type of chemicals or toxins that make it into the cell. So we have our rough and smooth. On the rough, because it is a double membrane system, it creates these spaces that exist, and those are called cisterns, like an empty jug, okay? So they're termed cisterning. And that's just the spaces in the endoplasmic reticulum. The Golgi apparatus looks like pita bread. All right. Once a protein is made by the ribosomes attaching to the endoplasmic reticulum, the endoplasmic reticulum, because it has a double membrane, it will wrap around that protein, transport it to the Golgi apparatus. The Golgi apparatus, as it begins to now move through all these flattened sacs of the Golgi apparatus, that protein is getting modified and packaged for transport. So by the time it leaves that last fold of the Golgi apparatus, that protein is now in its own membrane that makes its way to the plasma membrane and either continues and moves out, or the membrane fuses and spits the product out. Yes, no, maybe. Okay. The function of the Golgi apparatus is to accept from the endoplasmic reticulum a protein that is still not completely correct, get that product, move it through its own system, and by the time it's made its way through the flattened sacs, have a product now that can move to the plasma membrane and leave the cell. This would be an example, an example of this would be the digestive enzymes from the pancreas, mucus from the salivary glands, insulin from the pancreas. The product is now packaged and moves to go out of the cell. If there is a product made by the free ribosomes that stays inside the cell, it does get packaged. For example, lysosomes. Lysosomes are going to be a way that the cell can package digestive enzymes, but this time those enzymes, they might ingest a waste product inside the cell. They might help break down a substance that the cell needs. But one way that these become important, they actually, when the cell has done its job, it's no longer needed, it's at its end of its lifespan, it helps to digest the cell itself. The best way that I can explain lysosomes, have you ever seen children that are born, for example, where their two fingers might be connected together? And they have to go in and do the surgery to open the fingers up. During fetal development, the lysosomes didn't function properly. and didn't allow that